So we've been hearing about some therapeutic approaches and ways to improve protein therapies. I'm going to talk about how we can use micro and nanotechnology to improve the way we diagnose disease. And so we have this intriguing title, Putting the Squeeze on Cells to Diagnose Disease, and I'll get into what that means exactly. But I'd like to start out by kind of going through a process that a cancer cell might go through when it's starting in a tumor and invading into surrounding tissue and then going to other locations in the body where it metastasizes. And that's really what kills 90% of people that have cancer, is the metastasis. So we'd like to, certainly chemistry is important in this process, but we like to look at the physics of this process and understand more about how that plays a big role. And then eventually, if we can understand how physics is important, can we use some of our approaches to measure physical properties of cells and then connect that back to this disease? And so an example is a cell that's invading neighboring tissue, what we call the extracellular matrix, so this fibrous milieu of proteins that cells secrete around them. And so these cells have to kind of squeeze through this matrix, and so they have to be deformable, and they have to apply force to kind of pull their way through it. They have to squeeze then into, through this lining of our blood vessels that has both other proteins, smooth muscle cells in some cases, or endothelial cells, so cells that line the blood vessel, and have to get through these small pores and get into the bloodstream. Once they're in the bloodstream or in lymphatic vessels, other vessels in our body, they then have to get out in order to invade other tissue, right? So they have to kind of go through the inverse of this process and squeeze out into this new site. So these are processes where physics is certainly important, right? And so we're interested in studying that physics. And so I'd like to show a video of a balloon hitting a wall. So this is how we make use of our high-speed cameras in our lab. So it's a lot of fun. So this balloon is filled with honey. Interesting, right? So actually, if it's filled with water, it has all these crazy ripples and things as it hits the So one question I have for you is actually, how do you fill a balloon with honey? High pressure pump, that's a good idea. A funnel? A funnel is the starting point, or a syringe, over in the back. Right, so you want to push the air out while the honey is going in. Jeff, you heat it up, so that's a good, Jeff has found one of the solutions that, so my solution to this is actually hire some smart high school students like you during the summer, and so we did, and they found out the solution that Jeff figured out. So honey is very viscous, so we certainly have to do all the things that you guys mentioned, but we also have to heat it up to reduce its viscosity so that you can fill the balloon. Okay, so I don't just show this for fun. The reason to show this is that if you look at the dynamics of how this balloon deforms as it hits this wall, it tells you a lot about what's happening in the balloon. So it tells you actually about the viscosity of the honey that's filling it. It might tell you a little bit about the elasticity of the membrane that's inside of it. And these are things that we can see even without probing without looking with an X-ray or other things inside the balloon, right? Just by how it's dynamically changing in this environment, we can start to probe its internal structure. And this is important because cells actually have lots of internal structure that's important for how they interact with their environment, and for example, how cancer cells interact with their environment. What I'm showing here is a kind of a complex sort of structural distribution of proteins within the cell. And the cell has what's called a cytoskeleton, which helps to form its structure. Very similar to actually the way that, that some bridges and some architecture are built. So this is it's called tensegrity architecture, where you use lots of tension in different lines to kind of hold up something. And so a cell does a lot of this too. It creates this cytoskeletal structure that holds it up together. It also has a nucleus that's quite stiff, and the nucleus is filled with all this DNA that's packed to different amounts. And how much it's packed, that also affects how deformable the cell is. So these are things that are hard to see, but if you start to squeeze the cell, then you can start to see some of these internal structures. Right now, if we're going to analyze cells, we would normally analyze a cell by looking at the molecules that make it up, one at a time. And normally, what we'll do is we'll look for particular surface molecules on the cell, and we'll develop very specific probes that target those surface molecules, and then may make that cell light up if it has lots of those surface molecules. However, this doesn't tell us much about sort of the structural arrangement of proteins. It doesn't tell us how stiff that cell is. It doesn't tell us how that architecture of different proteins is laid out in the cell. And those are things that arise from this the assembly of all these proteins within the cell over time. Okay, so how do we start to look at maybe these structural aspects of cells that we think are important in disease? Well, we use a technology called microfluidic technology. The idea here is quite simple. We make very small fluid pipes. And we do this by leveraging the techniques from the computer industry that has developed ways to make very, very small features. So, the chip that's in your phone, in your pocket, all of those chips are made with the same technology that we use to make these microfluidic devices. Of course, we're not routing electrons, we're routing fluids in this case. So I'm going to talk to you about a specific microfluidic technology that we've been developing, which we call deformability cytometry, right? So the idea here is that we're making this measurement similar to that balloon, but at 10,000 times smaller scale, right? So a cell is about 1/10 the thickness of a human hair, 1/10 the thickness of your hair. And so we have to figure out how can we measure, how do we throw a cell against the wall and measure how it deforms, right? So that was the trick. And so I'm going to talk about how we did that. And so there's a few different pieces of this. One is that we wanted to align cells in a flow so that all cells were kind of following single file one after the other. They're all in the same location in the flow. We use a technique called inertial focusing. I won't be able to go into the details here, but it's a really kind of simple but kind of not obvious approach. The idea is that if you flow particles in a channel, that those particles that are randomly distributed coming into the channel don't end up randomly distributed coming out. They actually start to order and focus, and that depends on the shape of the channel. So this is like bizarre. When I first saw some of this, when I stumbled upon this, actually this was a serendipitous event when I was a postdoc, I was like, what the heck is going on here? This is ridiculous. Particles aren't supposed to do this, but they do, and you can control it pretty precisely. And so once these particles or cells are lined up, you can then stretch them in a uniform way. So basically, we have the cells that are lined up hit another opposing wall of fluid that's coming in the opposite direction, and then they stretch when they hit that fluid. Right? So boom, they hit the fluid, they stretch, and then they leave, and we can repeat this process. And we want them to be aligned so that each cell is kind of hitting the same wall of fluid. Then we use high-speed cameras. So we actually have cameras that can measure over 1 million frames per second. So how fast is the iPhone camera? Do you know how many frames per second is the iPhone camera? Right, 60 frames per second. This is 1 million frames per second. So 4 orders of magnitude more. Right? So we can measure very, 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 very fast things. So it's so fast that if you look at a bee, it looks like it's stopped. The bee's wings you can barely see them move. Okay. So this is the types of cameras that we're using. They actually use these, Mythbusters use these cameras. They use these cameras to look at explosions we're using them to look at cells hitting fluid and kind of exploding when they hit the fluid, I guess. Okay, so we take, this is great because we can measure many, many, many cells. So one thing that we know is if we measure one cell, that could tell us a little bit, but if we measure 100 cells, if we measure 1,000 cells, we measure 10,000 cells, that starts to tell us more about disease, because now we can see many different players in your blood, for example, or in another fluid where there's many different types of cells. So this is what the microfluidic chip looks like if you look from the top down. So you have cells that enter into this region up here. They then go around both of these channels. They hit what we call wigglers, which are ways to focus cells. And then they enter at the end this junction where they get smashed against this other wall of fluid, and we image with a high-speed camera. So what do the images look like? They look like this, or the videos look like this. Cells hit this region, they kind of hit here and kind of wiggle around and then leave, right? And then we extract information from these images. And actually, nowadays, we're actually using a lot of artificial intelligence and machine learning to extract all this beautiful information as the cell deforms and wiggles in that flow. So much information about the internal structure of the cell. And in each of these points is one cell. And then there's additional information in that profile, that structure of the population of cells. So this, and here we're extracting a few features from the cells. One we call deformability, which is this aspect ratio, how much it deforms. But there's a range of other features that you can extract from these types of images. For example, the dynamics of how the cell deforms. Okay, so we have this technique, and we started to look at it, using it in measuring disease processes. So I'm going to summarize some of this work in this slide here. So you saw this in the previous slide, you saw this kind of graph. And so on the x-axis, we have the diameter, the size of the cell. On the y-axis, we have this deformability. We did a study looking at fluids that build up around the lungs. They're called pleural effusions. This happens in patients that have pneumonia, congestive heart failure, but also like malignant diseases. They have cancer. And so fluid can build up because cancer cells are invading into that space and causing inflammation and other buildup of fluid around them. And so this is what we found is actually in patients that had malignant cells that we saw unique populations appear that were large in diameter and large in deformability. So that was kind of interesting. So we could see these malignant cells based on some of their physical properties. And they were different from other cells that line this lung lining, these mesothelial cells, which were large but not as deformable. And sometimes those cells were difficult for a doctor to tell the difference between. But what we didn't realize at the time, and it was kind of a surprise to us, beyond being able to see these malignant cells, is that we could see different states of the immune system. So we could see different activation states of white blood cells that were kind of also flowing out into this fluid. And we could see that in some states where there was a lot of these neutrophils that were activated, that neutrophils are involved in kind of the first response to bacteria or another pathogen invading you, that they look different, that they had kind of this more deformable but not much change in size. And then if we had sort of this chronic inflammatory process where you had lymphocytes and other cells that respond to like these chronic insults to our body, and develop antibodies, develop ways to target specific pathogens, that they had a very different sort of profile, this blue profile. And so there was, this is certainly not, there's some overlap between things, but we could start to see some differences just by looking at physical properties of cells. So why is that important? It's important because we can make this measurement very rapidly. And so in cases where we want to know, let's say a patient comes to the emergency room, we want to know right away, is that patient have a process where their immune system is very active because they have a bloodstream infection or sepsis, or do they have some viral infection, and maybe some of these lymphocytes are more activated. So these are the types of things that we can start to do very rapidly because we can measure them based on very simple mechanical changes. Okay, so to summarize, we're starting to bring this from some initial proof of concepts where we built this device. We started to make measurements here at UCLA, and now we're translating this out, or transitioning this out to a company that's going to bring it to the next level. And they're particularly interested at this finding that these white blood cells have very unique mechanical signatures that are associated with disease, and hopefully that can allow us to detect very early on whether a patient needs more aggressive therapy or needs to be admitted to the hospital right away, for example. Okay, so with that, I'd like to thank the team of folks that work on these types of projects in my lab, and if we have time for questions, I'm happy to take a few.